Assalamu alaikum. In this video, I discuss a checklist of the reading special figure. Special figures are included in your Viva and practical portion of the first professional MBBS examination, and these are the checklist. And according to this checklist, you have to describe this figure. So, at first you have to identify the form of the figure and justify your answer. So, these are the form of the figure: photograph, usual diagram. Uh, Within the usual diagram, realistic, semi-realistic, and schematic figure are included. You don't need to mention realistic, semi-realistic, schematic. Um, Identify this type of figure. Only say usual diagram. And there is the other type of diagram: low power photo micrograph, high power photo micrograph, transmission electron micrograph, scanning electron micrograph, composite 3D diagram. And this is the from. So this is the usual diagram, and justification of answer, because it is not taken by the camera. It is a drawing done by a human or by a computer software. So this is a usual diagram, and there is a no um, view of this figure. You have to identify the view of the figure. It's taken from front, back, or right or left. In this small uh, part of this figure is taken from the anterior lateral view is present here, but for this figure there is a no view is applicable. So there is a no view is here, and there is a no sectional view is present here, um, because in cell biology and histology, there is no sectional view is present. Only cross section of the outer portion of the cerebrum is drawn here. The principal issue of this here, the distributions of the nerves and neuroglia are drawn is this figure, and relationship between neuron and different neuroglia also seen in this figure. In this figure, we see a neuron, the processes of the neuron. This process of neuron is myelinated by the process of oligodendrocyte. I see the oligodendrocyte, a single oligodendrocyte is myelinate several uh, smaller portions of the nerve fiber, and its cell body is not present in the outer portion of the myelin um, myelin sheath. It is from the myelin sheath. And I also see the astrocyte. Astrocyte is line the outer portion of the blood vessels and take part in the formation of the blood-brain barrier. And this is the ependymal cells is line the ventricles, and this is the microglia cells, and these are the blood vessel. So by this figure we can understand the distribution of the neuron and neuroglia within the nervous tissue. Without this figure we cannot understand the myelination pattern within the central nervous system, how a astrocyte is take part in the formation of the blood-brain barrier, how a ventricles of the brain is lined by the ependymal cell, and the position of the microglial cell. So like this you describe this figure. So summary, you have to identify the form of the figure. This is usual figure because it is drawn by hand or by a computer software. This is the justification of your answer. In this figure, view is applicable, anterolateral view, and in this figure, view is not applicable. You have to mention this thing to your in front of the examiner. The view is not applicable for this figure, and for this figure, there is a no sectional view is applicable. You have to say this to get the number. And lastly, identification of the principal aspect. By this figure, we can understand the relationship between neuron and different type of neuroglia within the nervous tissue. And number 5, I describe what I see in this figure, and what thing I cannot understand by reading a text. um, Only I can able to understand the relationship between neuron and neuroglia within the nervous system by this picture clearly, but not reading a text. So this is all about video about spatial figure reading. If you find this video helpful, please subscribe my channel. Thank you for watching.